So in our presentation, we decided to look at the common squirrel. So we wanted to understand the squirrel's food preferences, behavior to predator odor, and reaction to obstacles in an urban setting. So we decided to choose Elm Park. So basically, we just wanted to look at the behaviors and preferences of urban squirrels in regards to competition or resource competition by determining their food preferences, how they behave with their food in the presence of a predator, and the lengths that they will go to obtain that food. So this formed our three questions, which are what are the food preferences of squirrels within an urban park? Will squirrels decrease foraging behavior if coyote urine is applied to the food source? And then lastly, will squirrels at Elm Park retrieve walnuts less frequently if they are blocked by various obstacles? So just doing this allows us to develop an understanding of the common squirrel and how they react to the changes and challenges of their resources in this urban setting, along with observing and recording how far they will go to obtain these resources. So in our first experiment, we did a cafeteria experiment. So our hypothesis is just that there will be a difference in food type preference of urban squirrels. So we chose four different types of food. The first one is sunflower seeds, apple slices, walnuts, and then cornflakes. So in the methods, we did 10 trials each of four of the four different types. So that gave us a solid 40 data points. Um, at the beginning, we weighed 10 grams each for all the plates so that they had a nice baseline. And then we placed them in a square where they were equally distant from each other, just like we did in lab. And then we put them throughout Elm Park. But I decided to do two sets of 5 trials each just so that it could be more spread out and not just like stacked on top of each other. And then we left them for 60 minutes. And then when I came back, as you can see in the bottom right corner, some of the food had spilt out on a lot of them. So I had to pick up all the little pieces and put it back in the tray because technically that's food that isn't eaten. So it still needed to be part of our measurement. So I tried to pick up all the little pieces, put it back in, then I would weigh how much was left and I would divide that weight by the initial 10 grams to figure out what our percent eaten is for all the foods. And then when I had all of the data, we did an ANOVA. So basically, an ANOVA just asks if there's a difference between the means of the percent eaten. So our null hypothesis is that there is no difference in means between the percent eaten of all the food types. So basically, the mean of the walnuts equals the mean of the sunflowers and etc. Um, but the alternative hypothesis states that there's at least one difference in means between the percent eaten of food types. So maybe the walnuts don't equal the sunflowers, or the sunflowers don't equal the apple slices, but it doesn't really matter which one. It can be any combination. It just means that one of the means does not equal another one. But our null hypothesis was aggressively rejected with our very small p-value of 4.31 times 10 to the negative 6. So this means that we could conclude that there is a difference in food preference, or there is a food preference between at least one food type. And this is a box plot that just represents all of the data. So we did know that there was a difference, but we didn't know between which foods there was a difference. So in order to figure this out, we did a Tukey's post hoc test, which just compares all of the pairs of means to find which ones are actually significantly different. So this gave us 4 significant p-values. So if there are 4 pairs that are different from each other, so the sunflower is different from the apple and the cornflake, and the walnut is different from the apple and the cornflake, but the sunflower and walnut are not different from each other, and the apple and cornflake are not different from each other. So what does this really mean? The sunflower and walnuts were different from all the food types besides each other, and this was kind of expected because the squirrels seem to prefer the food that resembled the resources they're more likely to find in nature, so a nut and a seed. But when we were trying to find data to back up our experiment, there was a study that found that the frequency of squirrels can change, the frequency of them eating can change seasonally. And they found the peak consumption frequency occurs in the fall, which would make sense because they're going to stock up for the winter and take more food in the fall for that season preparation. So this could play a role in our data because there could maybe be no difference between the sunflower and the walnuts, but or it appears that there is no difference because maybe they're just taking more food to stock for winter. But in reality, maybe if we tested in summer, there would be a preference between the sunflower and the walnut. So why is this even important? So the study that we found kind of backed this up because it compared the eastern gray squirrel as an introduced species to the western Douglas gray squirrel in Washington. So basically they just looked at the diets of each of the squirrels and just wanted to understand the competition between these and how their resources overlap. So it's just important because as we learned in lecture, competitors can coexist if their resources are just different enough, which is exactly what they found. So it's just important to understand because in times of stress, there could be more competition interactions because they do have that little bit of overlap in their resources. And then we also found that ground squirrels can vary their diet depending on age. So juveniles may have a different diet composition than adults. Adults give birth in the early summer and the late fall. So it could have been that some juveniles were present in our experiment and taking different food resources than adults, and maybe that just affected our data a teeny bit. And then lastly, limitations. Some of the limitations were that it was hard to spread the sites on multiple squirrels wouldn't hoard one site because sometimes one squirrel would see another squirrel eating and then there would be like 4 of them at one site and none at a different site, so that could have affected our data also. And then the weather played a role because sometimes the plates would flip over and then I would have to pick up all the little pieces and put them back in, and it just could have affected how much was actually left in the end. So that is the conclusion for our first experiment, and now Allie will do the second one.
Okay, so for experiment number 2, it is based on the foraging behavior of squirrels in the presence of a predator's odor versus without. The larger question at hand being, will squirrels decrease foraging behavior if coyote urine is applied to a desired food source, such as walnuts? And this overall experiment is inspired by the Mueller-Schwartz experiment in 2009. My hypothesis originally being, if coyote urine is applied to the walnuts, then the squirrels will not retrieve them. My methods are listed below, and I had used white coffee filters, a 5 milliliter plastic pipette, California walnuts that were whole shell that I had purchased from Amazon, as well as coyote urine, which was generously provided by Professor Mathis. Our location was Elm Park, located in Worcester, Mass., as most of us have heard of it, since it's not too far from Clark. In terms of duration, I had visited Elm Park a total of 4 times. For setup, 4 coffee filters were placed in a square shape with 1 walnut per coffee filter. There were 2 treatments total, walnuts with the urine applied and walnuts without. 20 trials were done for each treatment with a grand total of 40. Each square, I like to call it, was placed at the base of a tree, and these squares, or the trials themselves, were randomized around the park. This meant that squirrels wouldn't necessarily associate a certain treatment in a certain area or with a certain tree, and that would skew the data as I had predicted. So I tried my best to randomize it as best I could, and each trial was timed at about 45 minutes, and this allowed for ample time to observe the squirrels and monitor the walnuts that were being taken or not taken. As I had already mentioned, this experiment is inspired from a pre-existing one, Mueller-Schwartz 2009. I originally had used 2 wooden grids to keep track of the walnuts, and I had to modify this original method since the wooden grids actually prevented the squirrels from taking the walnuts in a natural way and restricted the experiments time-wise. So instead I modified it from the left picture at the top to the bottom left picture, where I had just used 4 coffee filters, as I already mentioned, and I would hold them down against the wind with barks and twigs, and this actually proved pretty successful, and you can see that a little squirrel on the right is actually eating one of the walnuts. So for my alternative and null hypotheses, again with the overall question being, is there a significant difference in means between the number of walnuts removed and the treatment type? For the alternative hypothesis, there would be a significant difference in the means, and for the null hypothesis, there would not be a significant difference in the mean. For my results, I used a Welch two-sample t-test in our studio with or I should say with my results that are on the right in parentheses. I had obtained a t-value of negative 1.2624, and this actually indicates the reversal in the directionality of the effect, and this had no bearing on the significance of the difference between the treatment types overall. A degree of freedom of 38, and more importantly, a p-value of 0.2145. Figure 1 depicts the box plot that I had created in our studio, and you can see the two treatments on the x-axis labeled with odor and without the predator odor, as well as the walnuts that were removed between the numbers 0 and 4. The general trend we can see is that the mean and median of the walnuts removed without the odor were greater than those removed with the odor. I had additionally made histograms to depict these trends. These histograms actually combine both treatment types, so they are not distinguished here. They instead display an overall pattern or trend that I saw. So figure 2 depicts the histogram that displays a frequency of the number of walnuts that were removed. And we can see that 4 had the highest frequency of removed walnuts. So this means that in more cases between both treatments, 4 walnuts of 4 were removed total, and this means in figure 3 with the walnuts remaining that there would be a higher frequency of 0 remaining if the most being 4 were removed. This also means that a lower frequency of walnuts removed and remaining were between 1 and 3 walnuts. It was an all or nothing scenario in which a squirrel would either take all 4 or not take any at all. I thought it would be important to discuss that predator odors can have various behavioral effects on prey, such as their rate of activity, decreased or increased foraging, and physical movement to a safer habitat if they feel threatened. This is backed up by Hegab in 2015 on their study. Based on the figures that I did generate, the number of walnuts removed with the odor present was not significantly different, and this shows in my p-value, which was 0.21, and we know this is greater than 0.05. Therefore, my alternative hypothesis is rejected and my null hypothesis is accepted, meaning that there is no significant difference between the mean value of walnuts that were removed with or without the odor. Overall, there was a greater mean number of walnuts taken for the treatment without the predator odor, and this was what I had originally predicted, but this was not significant. And a higher frequency of squirrels removed all 4 walnuts regardless of the treatment, so there was also a higher frequency of 0 walnuts remaining. So one might wonder why the coyote urine would not be significant when it comes to whether or not the squirrel will retrieve the walnut. Potential reasons being that coyotes aren't common predators around Elm Park since it is an urban setting. It's possible that there were a mismatched predator odor and the squirrel prey species, and this could have to do with its repellent effects. Elm Park squirrels, as most of us know, can be ruthless and determined. They're not really afraid to go after what they want. And Parker and Nylon did a study in 2008 that further emphasizes this and their aggressiveness overall. So it could also just be that the squirrels were just very determined to get what they wanted, and that would be their food source, the walnut. Limitations of the study include that I had utilized the same coyote urine throughout, which likely aged over time and wouldn't have the same pungent effects as a fresh sample. And experiments were weather permitting, and the squirrel activity did depend on the time of day. As I already mentioned, I had visited Elm Park on 4 separate occasions, and the weather was different, and I did go at different times of the day, so this could definitely be a limitation to the experiment overall. Future directions would be making sure I do collect my data at the same time of day and making sure the weather is more or less the same for more accurate results. And I would conduct possibly the same experiment at a rural park, still using the coyote urine to see the results I would obtain. I could use a native predator or a climbing predator's urine instead, and this could possibly pose more of a threat to the squirrels if a climbing predator's urine was used. They just might have a heightened sense of fear if this were the case. And I could also use the fur of a predator instead of urine. Maybe a visual that the predator was there recently could have more of an effect on the squirrel's foraging behavior. And that is experiment number 2, and Athena will be discussing experiment number 3, which has to do with foraging behavior and obstacles.